Hi, my name is Reagan, and today I'm going to be dissecting a monocot flower and a Eudicot flower. My Eudicot flower that I'm dissecting today is a rose, and my monocot flower that I am dissecting today is an astromeria. Monocot and Eudicot flowers vary in different ways. Some differences are their seed structures, their leaf vein structures, and also their vascular bundles and the way those are formed. When monocot seeds germinate, they form one cotyledon. When Eudicot seeds germinate, they form two. The veins in a monocot leaf run parallel, as you can see in the astromeria's leaf. Veins in the Eudicot leaf run branched off of one main one, as you can see in the rose's leaf. The way the vascular bundles form are also different. The monocots form scattered-like, while the Eudicots form in a ring formation. So this is my monocot flower, this is the astromeria, and this is the stem. It provides support of the flower and it sends water and nutrients to the flower and the leaves as well. This is the leaf. Can't really tell on this one, but the veins in this leaf run parallel. And the leaf provides food by using photosynthesis. This is the flower, the astromeria. And right here where my finger is, this little section right here, this is called the ovary. So now we are left with just the flower. And as you can see, we have two different types of petals here. So the outside ones that do not have any pattern on them, these are called our sepals. And they are to protect the flower bud as it's blooming. They usually come in multiples of 3. So I'm going to pick them off here. 1, 2, 3. So here are all of our sepals. And now we're left with just the petals, the flower petals. So what we're left with is the internal flower, and what we see right now are the male parts of the flower. This little part right here, these little dark green things at the top, these are called our anthers, and this is where the pollen is produced. And then the stalk that is on it, that's called our filament, and that supports the anther. And now I'm just pulling off the male reproductive parts so I can be left with the female reproductive part called the carpel. And the carpel is made up of the stigma, which is where the germination happens, it's the very tip. And then the stalk is called the style, and that just supports the stigma. And of course, we have our ovary, which, as I said before, carries our ovules, which will soon turn into seeds. So what we're left with the astromeria is the ovary, and that contains the ovules, as I said. And I can cut it in half to try to show you the ovules here. So as you can see, there's a lighter green in the middle, that is where our ovules are. And those will soon develop into seeds to start some reproduction. The Eudicot flower that I'm going to be dissecting today is a rose. So here's the stem, and as I said before, it provides support for the flower, and it sends water and nutrients to the leaves and the flower. Here are the leaves. Eudicot leaves have one main vein that branch into many different ones, so they're called branching veins. And then the flower is the rose. The flower consists of the sepals, which are these green ones right here, and then the petals, which are right here. The sepals protect the flower when it's budding. And then the petals, which are the red ones, they attract the pollinator. So I went ahead and I tore off our sepals and our petals of the flower, so we are left with the reproductive parts of the flower. The purplish, pinkish parts down here, those are the male reproductive parts of the flower. And in the middle, the pinkish yellowish parts, those are the female parts of the flower. The male reproductive parts is called the stamen, and they consist of the anther, which creates the pollen, and the filament, which supports the anther. And if I tore all of these off, I would be left with the female reproductive parts, which consists of the stigma, the style, and those all lead into the ovary. We will get to that in a second. And now we're left with our female reproductive parts. That consists of the stigma, the yellow spikes at the top here. That's where the germination process happens. And then the brighter pinkish parts, those are our styles, and they support the stigma. And they all lead into right here, which is called our ovary, which again has our ovules, which soon turn into seeds. So I cut the rose ovary in half to expose our ovules, and here you can see them really nicely. The ovules will soon turn into seedlings.